Very straightforward. You're going to learn some transfer techniques that will allow you to move microorganisms from one sterile environment into another sterile environment. This is something you will have to routinely do in lab, particularly when you're working with your unknown. You need to take things out of your unknown tubes to do tests. You don't want to contaminate it in the process. And you constantly need to subculture your unknown so that you always have a viable culture to work with. So these transfer techniques are really important. So basically what you're going to do is you're just going to practice transferring from a variety of different types of environments to others. So the major kinds of things you're going to be working with tomorrow will be broth tubes. And so you will be transferring from a broth culture to another broth culture. You'll be transferring from a broth to a slant culture, from a slant to a slant, from a plate to a slant, or a plate to a broth. And the techniques really need to become just kind of second nature to you. And you'll do them enough that that will be the case. But this is just kind of fundamentally important things. A word of caution. With the broth tubes, remember, these caps are push-on caps. You're not screw-on caps. So if you do not keep these tubes upright, they will spill. And I can pretty much guarantee that there will be at least one spill tomorrow. If that happens and it's small, you can use just the Clorox and paper towels to wipe it up, put it in the autoclave bag. If it's big, let Dr. Morton know, and we can use some of that powdered Biosorb to soak up a lot of liquid. A couple of things just I will reiterate that he'll probably tell you as well. As the way we're going to sterilize our transfer instruments, you'll use a loop tomorrow primarily, is in the flame with the Bunsen burner. So turn on your Bunsen burner, and then you need to insert the loop into the Bunsen burner flame, and it should turn red hot. So you should be able to see it turn red. Initially, or ideally, you will also make sure you pass this portion, the shank of the device, through the flame as well, because a lot of times this is shorter than the depth of the tube, and so sometimes you'll get the shank in there as well. So it's not a bad idea. This doesn't have to turn red hot to make sure it passes through the flame. Okay. Once you have sterilized these, don't set them down. If I do this now, it's unsterile. Okay. If you need a hand, what you can do is you can set it on something like this. Yes, there is a finite chance that a bacterial particle could alight out of the air onto the surface of this, but it's not that likely. Okay, so try to keep these off the tabletop when you use them. When you're transferring from tubes, there is a tendency to want to do this when you uncover it. There's a couple of things wrong with that. Number one is that's going to leave this tube open longer and give a greater chance for contamination. And when we set it down here, yes, this is not touching, the open side isn't touching the non-sterile surface, but things could fall in there, okay? So what you really want to do is, with whatever your dominant hand is, you'll have your inoculating needle or loop in one hand. Take your little finger and pull the cap off like that, and you never set it down. You'll do your transfers with this cap in your little crook of your little finger, okay? Before you go into the tube, you pass this through the flame of the Bunsen burner to sterilize the neck, and you can just kind of rotate this in your fingers to get all surfaces. Then you'll go in and make your transfer, okay? Don't set this down now. Reflame. Now you can put this cap back on again, and then you can go over and take the second one off, reflame, introduce this in here, streak it on the slant, slosh it around in the broth, reflame again, put your cap back on, and then finally re-sterilize this, because there will be some stuff left over on that needle or loop. Okay? There's a tendency to want to do these one at a time. I would encourage you to try practicing first with empty tubes, so that you're having both tubes in the offhand at the same time. And if you hold them in the palm of your hand like this, then you can move them around if you have some practice, and you can sometimes work with these simultaneously. If you get good, you can do 3 tubes in one hand, so you can transfer from one tube to 2 other tubes. But try to get out of the habit of doing this, sitting like this, pulling this one out, doing this, setting this down. It's a lot slower, okay? So I would recommend tomorrow that you spend a little bit of time just go grab a couple of empty test tubes with caps on them and put them in your left hand if you're right-handed, and start just getting comfortable manipulating these, taking the caps off. Okay. It becomes second nature to you, but tomorrow you'll just do some practice with that. There are self-explanatory diagrams in your lab handout that show you step by step the things that I just illustrated. The other thing that the lab does ask you to do is to evaluate your plates from last time. Clean and dirty countertop and wash and unwashed hands. I was just looking at them, and at 25 degrees Celsius they grow pretty slow. So I think what we're going to do is just have you leave those plates over the weekend in an incubator, give them a chance to grow some more nasty stuff. And then on Monday, if you'll remind me, we'll have you take those out of the incubator, put them in the refrigerator, and then in Wednesday's lab, you can then do that follow-up evaluation. Next Wednesday, you will learn to do another couple of important techniques. One is a streak plate, and then the other is a pore plate for motile bacteria. And you're going to get your unknown. So we'll talk a little bit about the unknown and spend some time talking about kind of the expectations for that semester-long exercise and get you started on that process. Okay? Any questions? Okay. So we were in the midst of talking about the cell envelope. Anybody remember what the two components of the cell envelope are that we're going to talk about?
Cell wall and cell membrane.
Cell wall and cell membrane. What is the structural component that is present in all bacterial cell membranes? Found in both gram-positive and gram-negative. Peptidoglycan. Peptidoglycan is a polymer. It's a chain of repeating subunits. The repeating subunits that make up this substance, peptidoglycan, is called glycan tetrapeptide. So peptidoglycan is a bunch of glycan tetrapeptide units stuck together. On this diagram right here, it shows you the chemical structure of glycan tetrapeptide. I want to say some things about the various components of that unit. I do not expect you to be able to draw structures. I am not a big person of being able to reproduce structures from memory. You can look those up. But I do think it's important to know what glycan tetrapeptide consists of. And particularly, there are some unique properties of glycan tetrapeptide that I want to concentrate on. There are two sugar molecules that are found in glycan tetrapeptide. And both are modified forms of glucose. But they're sugars you probably haven't really ever heard much about before. They are abbreviated NAG and NAM. NAG stands for N-acetylglucosamine. You see the word glucose in there. You see this amine indicating that this is a nitrogen-containing sugar. Okay, so there's some nitrogen functional groups that are attached. NAM stands for N-acetylmuramic acid. Now, again, if you're curious and you kind of want to see what these things look like and how they differ structurally from glucose, then that's the purpose of this particular diagram up here, is to show you the structure of those things. Okay? Those sugars are covalently bonded to one another to form a carbohydrate chain. So up here in this particular diagram right here, The purple and the yellow represent the alternating N-acetylglucosamine and acetyluramic acid components. That chain is covalently bonded to this chain by this complex bridge of amino acids. Sometimes that's referred to as the interbridge. So this would represent peptidoglycan as a whole, and peptidoglycan consists of these repeating units of 2 sugars and then this complex inner bridge amino acid structure that can link one polymer of sugars with the other. The way in which these particular chains are bonded to one another ultimately determines the overall structure of the cell wall and consequently the shape of the cell. Now, if you look at this inner bridge in a little bit more detail, you'll notice that there are several amino acids that probably sound familiar to you, like glycine and lysine and alanine. But you also see these designations of D and L. And those, everybody said organic, right? What does D and L stand for? Dextrorotatory and levorotatory, okay? The isomeric forms of the amino acids. Does anybody know which form of amino acid, D or L, tends to predominate in most proteins in most living organisms? No. Now, natural selection, for some reason, has tended to favor the L forms of these isomers of amino acids. What's unusual is that we find D forms of those amino acids in this inner bridge region. So that is something that is a little bit unique to peptidoglycan, something we don't see very commonly. Sometimes in this inner bridge region, although it is not indicated here, we will also see some unusual amino acids that you probably haven't ever heard of before. One that is not uncommonly found in the inner bridge region of peptidoglycan is a substance called DAP. That is diaminopimellic acid. And this is about the only place that you really find this particular type of amino acid is in this peptidoglycan region. So there's some kind of unique chemical composition that is found in peptidoglycan that we typically don't see anywhere else. Okay, let's go back to these slides that we've seen previously. What is the major distinguishing feature between a gram-positive and a gram-negative cell wall? Gram-negative has less peptidoglycan. If you do it on a percentage basis of the entire cell wall structure, typically we see maybe about 15 to 20% of the cell wall mass in a gram-negative composed of peptidoglycan. In the gram-positive cell walls, it's really more like 80 to 90% of the mass is this peptidoglycan stuff.
Gram-negatives have the outer membrane.
Gram-negatives have that outer membrane, that LPS layer. And the LPS layer has some interesting properties that we'll talk about in just a second. So, here's what a typical gram-positive bacterial cell might look like. So down here for reference, this is the cell membrane right here. And then fairly tightly pressed up against the cell membrane is this thick layer of peptidoglycan. Interspersed within this peptidoglycan is a molecule called teichoic acid and sometimes a lipid derivative of that, lipoteichoic acid, and they can be interspersed in and among that peptidoglycan layer. These molecules tend to have negative charges at physiological pH, and they tend to contribute to the overall negative charge that most cell walls carry. Different books will disagree with this next one. In this particular slide, they illustrate a little space here between the cell membrane and the peptidoglycan layer. And that little space is called the periplasmic space. If it's present at all, it is very, very narrow. Some books will tell you that there is no periplasmic space in gram-positive bacteria, that this cell wall is actually just pressed right up against the cell membrane. Other textbooks will disagree, as this slide did, and say there is sometimes this little space in here between the membrane and the wall. We'll talk about what the periplasmic space does when we talk about the gram-negative bacterial cell wall in just a second. Peptidoglycan isn't really much of a barrier to entrance of molecules, and so it really doesn't have a permeability regulatory process. Instead, it is primarily intended to maintain the shape of the cell and to maintain its integrity so it doesn't burst when it's present in a hypotonic environment. That's its main function. Yeah, peptidoglycan isn't really a permeability layer. It's really just intended to kind of maintain cell shape and protect the cell from bursting. Okay? Gram-negative cell wall. Okay, so here's our cell membrane. Here's our thin layer of peptidoglycan. And then here's our second layer, the LPS layer. In gram-negatives, we definitely have a periplasmic space. Okay, so between the peptidoglycan layer and the cell membrane is this space called the periplasmic space. One of the things that tends to happen is that enzymes accumulate in that space. And oftentimes those enzymes are involved in digestive processes. One of the things we didn't really talk about a lot the first day of class when we talked about the roles of microorganisms is that microorganisms are great decomposers. Okay. Typically, however, that involves secreting an enzyme into the environment where those molecules are present that are being decomposed. Now, if you just secrete them into the environment, those enzymes could be lost. But what typically cells will do is they will secrete those enzymes into the periplasmic space. And the peptidoglycan layer kind of helps to hold them in there and retain them. Then molecules from the environment can move across the cell wall and into the periplasmic space, and that's where the digestive processes can occur. Periplasmic space is where you also oftentimes find the receptors that regulate flagellar rotation. You may or may not remember I said that there are receptors out there that can bind to chemicals, and depending upon what receptors are bound, that can influence flagellar rotation, being clockwise or counterclockwise. Therefore, you get runs and tumbles, and that determines whether your chemotaxis is attractant or repellent. The LPS layer is a lot like a cell membrane. It is a phospholipid bilayer. Embedded in it are a variety of channel proteins. called porins. Remember, cell membranes are notoriously impermeable to a lot of things. They help to regulate what can enter and leave the cell. And this outer membrane, the LPS layer, has that function as well. Some things can pass, some can't. Well, if you want to get a large molecule out of the environment into the periplasmic space so it can be digested, that large molecule is going to have a tough time moving across that phospholipid outer layer. Those channel proteins provide a mechanism to move large substances across that layer. We're going to talk later on about antibiotics. The first antibiotic that was discovered was penicillin. And there's several different kinds of penicillin. We'll talk about that as well. But the naturally occurring form of penicillin, penicillin G, is only effective against treating gram-positive bacterial infections. So it's really good at treating a staph infection because staph is a gram-positive bacteria. But it will do a lousy job, if not being totally ineffective, at treating gram-negative infections like E. coli infections. Why? Well, what we're going to see later on is that penicillin interferes with cell wall synthesis. That antibiotic has to get access to the peptidoglycan layer to inhibit its resynthesis. The LPS layer is impermeable to naturally occurring penicillin. So over the years, one of the chemical modifications that was made to penicillin in the lab was to tweak it a little bit. So we've now got a synthetic form of penicillin that this outer layer membrane is permeable to. Now that form of penicillin will treat both gram-negative and gram-positive infections. It is moved by that modification from being what we call a narrow-spectrum antibiotic, only affecting certain kinds of bacteria, to becoming a broad-spectrum antibiotic, affecting both gram-positive and gram-negative. Embedded in this outer membrane are some lipopolysaccharides. And that's the source of the name LPS layer, lipopolysaccharide layer. And one of the components of lipopolysaccharide is a molecule called lipid A. So lipid A is a component of a lipopolysaccharide molecule. Lipid A is an interesting substance. When gram-negative bacterium die, their LPS layer breaks down. So does their cell membrane. But when the LPS layer breaks down, this lipid A component is released from the LPS layer. And unfortunately for us, this substance can be toxic. It typically won't kill you, but this particular toxin can affect dramatically the permeability of epithelial cells, particularly of your gut. And all of a sudden, water can come rushing out of the tissues and into the colon when previously it couldn't. So the permeability of those cells has changed. And if you lose too much water to your gut, at the very least you suffer from diarrhea. You're probably going to be vomiting because you want to try to get rid of this toxin. But in severe cases, you can lose so much fluid from the blood that you can go into hypovolemic shock. And you could potentially die from that. Adults, very little chance of this happening. But a young child or a baby who gets a big dose of lipid A toxin, it could be fatal for them. This particular type of toxin is called an endotoxin. And we'll come back and talk about this later on in more detail.
Do the cells, like you said, it's when the bacteria cells die. So like, if an antibiotic were to come in and like kill it, does that?
Bingo, absolutely. And that's one of the concerns about using antibiotics that are broad spectrum. Because if your infection is a gram-positive staph, for example, but you take one of the more broad spectrum antibiotics, if you've got a lot of gram-negatives also associated with that infection, then the antibiotic can kill them as well. You can actually release this endotoxin as a result of death of those cells, and so there can be complications associated with that antibiotic therapy. Now, fortunately, short doses of 7 to 10 days typically don't cause a problem. But it's something a physician does have to be aware of is, could your treatment cause a secondary condition like this endotoxin poisoning? There is a species, a genus of bacteria, that is gram-negative, that is notorious for releasing endotoxin when it dies, and that is Salmonella. Anybody ever suffered from salmonellosis before? Salmonella food poisoning? It's not pretty. Okay? That endotoxin really affects your digestive tract, nausea, vomiting. You have to really make sure you keep hydrated, otherwise that can be problematic. And although it's not going to kill you, sometimes you're so miserable that for a while you feel like, you really wish you could die. So this can really be a very unpleasant kind of thing. So here is a kind of a comparative chart that shows you some of the differences between gram-positive and gram-negative bacterial cells. Number of major layers, 1 peptidoglycan, basically. Here, small peptidoglycan layer than the LPS layer. Chemical composition. Notice here over here the lipoproteins, the porin proteins, the lipopolysaccharide. Overall thickness, outer membrane, yes, and gram-negative, no here. Very narrow, if absent, periplasmic space, much more extensive periplasmic space. And then relatively permeability to molecules, because you have the second membrane, molecules tend to be less permeable to the gram-negative cell wall than the gram-positive cell wall. Okay, gram stain. Something you will be doing in 2 weeks, I believe. Fundamentally important staining technique that will begin to divide all the bacteria into one of two groups, either gram-positive or gram-negative. This shows you the outcome of the gram staining procedure when it's finished. Gram-positive bacteria are bluish purple. Gram-negative bacteria are a pinkish color. So why do we get different outcomes from the same process? It's a reflection of the fundamental differences in the cell wall structure of those two bacteria. So let's run through the gram staining procedure right now and kind of see what's happening to the dyes in the cell wall as you go through the various steps. Gram stain is a type of stain called a differential stain. As that name implies, the stain differentiates one cell type from another. Differential stains have more than one staining reagent associated with that process. There are some stains called simple stains which only have a single staining reagent involved. You'll learn how to do both simple stains and differential stains. The first step in the gram staining procedure is, after you get some bacteria on the slide and let that slide dry, because typically you're transferring into a drop of water or a drop of broth onto a slide, you let the water evaporate. You can hasten that a little bit by putting it under a gooseneck lamp, for example. But once it's dry, it'll look something like this. You can barely see if you look at this, a little kind of opaque region on the slide. And one of the mistakes students typically make when they make slides is they put too much growth on the slide. And then this is not just a little thin opaque region, but it's really just totally cloudy. And it's very hard for the stain to penetrate into that. And in gram stain, it's very hard when we do this decolorization process for the alcohol to consistently decolorize these slides. So don't put too much growth on it, okay? The next step in the slide process is using your little forceps. You just pass this through the flame of the Bunsen burner. That's called heat fixation. And that will kind of adhere the bacteria to the slide so it's not washed off during the process. And then once that's done, you apply the primary stain, which is a purple-violet stain called crystal violet. And you let it soak on the slide for about a minute. And then you just rinse with water, and when you're done with that particular step, the cells will have absorbed the stain. Crystal violet carries a positive charge. And remember I told you the bacterial cell walls have a negative charge, so the stain will bond to the cell wall. So after the first primary stain is applied, both gram-positive and gram-negative cells will look purple. Okay? So we haven't differentiated yet. The next step in the staining procedure involves flooding the stained slide with iodine. The type of iodine we use is called Graham's iodine. Graham developed the gram stain. Iodine serves as a mordant in this particular procedure. Does anybody know what that term means? Sounds a lot like the word mortar, doesn't it? Ever heard of mortar before? What's mortar do?
Sticks them together.
Yeah, it sticks stuff together. It's kind of a cement. What happens is the iodine binds to the crystal violet and forms a crystal violet iodine complex that is quite insoluble. So it kind of binds the iodine. You then rinse off the iodine.
Then why is there no effect on the gram-negative if we're still stained?
Because in this particular case, in both cases, John, we've had stained cells that have now just been kind of bound with iodine. The color will not change because the crystal violet iodine complex is still purple.
But it says on the right in the chemical reaction that there's no effect. Why is that? If there's still like the...
I would disagree with that statement, to be honest with you, because there is an effect with the iodine, but because this layer is so thin, it's a minimal effect compared to the thicker layer of the... because what's happening is the crystal violet's binding to the peptidoglycan layer. Okay. So if we look at these cells at the end of the iodine stain, they still look the same. They're still purple. The key step in the gram stain now is decolorizing. And that's accomplished by rinsing the slide with alcohol. This normally insoluble crystal violet iodine complex is actually fairly soluble in alcohol. It's not washed out with water, but it's pretty soluble in alcohol. So as we start rinsing those slides now with alcohol, you'll see purple stain come bleeding out of the preparation. The key is to decolorize for the right amount of time so that you wash the stain out of the gram-negative cell wall, but not the gram-positive cell wall. Is that hard? No, not really. Because again, if you look at the thickness of the peptidoglycan layer, if you rinse for about 10 seconds, and that's kind of variable depending upon how thick your smear is, but if you decolorize for about 10 seconds, that's sufficient to wash out all of the crystal violet iodine out of this thin peptidoglycan layer in the gram-negatives. But it's not a sufficient amount of time to completely decolorize the thicker peptidoglycan layer of the gram-positives. Following that decolorization, we rinse with water. So if we were to look at those cells now, gram-positives would be still purple, because they would have retained some of the dye, but gram-negatives would be colorless. Well, it's really hard to see a colorless bacterial cell on a slide. So the last step involves a counterstain. And the counterstain that we use is a red dye called safranin. And it binds to both cells. But if you bind a pinkish red dye to an already intensely stained purple cell, it's still going to look purple. But if you bind it to a colorless cell, that's now going to take on that pinkish purple color. So you rinse off the counter stain, blot the slide dry, look at it under the microscope, and gram-positive should be purple, gram-negative should be pink. So the key is don't over decolorize. Because if you do, you can eventually wash out even the stain from the gram-positive cell wall. Well, that's going to be pretty critical with your unknown, right? You want to make sure you get the right gram reaction. So if I just put a bacterial smear on a slide and go through the gram staining procedure, and it comes out to be gram-negative, can I really be confident that that really is truly a gram-negative bacteria, as opposed to maybe I just decolorized too long? I can't. How could we mitigate against making that mistake?
Take a known gram-positive liquid.
Yep. Ideally, take both a known gram-positive and gram-negative, put them on the same slide, so you'd actually have 3 smears on the same slide, and then when you treat that slide with the gram staining procedure, all of those smears should be equally exposed to the same conditions. And if the positive control comes out gram-positive, the negative control comes out gram-negative, then you can be pretty confident in the outcome of your result. So that's something that we will continually do, is run controls on your slides for gram stains. Now, once you get good, you can really kind of tell, once you see that purple color just stop leaching out of the slide, you can stop. It might not take 10 seconds all the time. And as a matter of fact, when you do this on the lab exam, you will have to do this. There'll be a set of stations set up right where John and David are sitting, where you've got some cultures, some slides, some gram staining stuff. You're going to have to do a gram stain. And typically, we don't give you controls to do that. You've done it enough by that point that you can be pretty confident in the outcomes. Now, not all cell walls are exactly like we just illustrated. The vast majority of bacterial cells have a cell wall. But there are some bacteria that entirely lack a cell wall. mycoplasms are notorious for having no cell wall. Anybody ever heard of walking pneumonia? Ever had walking pneumonia? Not pleasant. Walking pneumonia is typically caused by mycoplasmic bacteria. In fact, sometimes it's called mycoplasmic pneumonia. Mycoplasms tend to be highly pleomorphic. And that actually is illustrated in this slide right up here. Whoops. Look at the diversity in the shape of these particular cells. Some tend to be more rod-like. Some tend to be more kind of cocci-like almost. Sometimes they form branches. They're just kind of all over the place in terms of their cell structure. How come?
They don't have the rigidity of the wall.
They don't have that cell wall that normally determines shape. What's the downside of not having the cell wall?
Desiccation would be bad.
Not so much desiccation, because desiccation would involve the water rushing out of the cytoplasm. It would be,
Things can easily go into the cell.
yeah. I mean, if too much water enters the cell by osmosis, then the cell will burst. Mycoplasms are fairly unique in that in their cell membrane we find a fair amount of steroid molecules. Now that's not unusual for eukaryotes. Remember, we have cholesterol in our membranes. Sterol molecules tend to give membranes more rigidity. Well, if you don't have a cell wall, it would be nice to have at least a fairly tough cell membrane. So the presence of these sterols is really a consequence, probably, of the lack of the cell wall. There are another set of interesting organisms in terms of cell wall structure that are called L-forms. L-form bacteria can lose their cell wall entirely during certain stages of their life cycle. Sometimes they have a cell wall, sometimes they don't. If you happen to see an L-form in its no cell wall life stage, what gram reaction would it have if you gram stained it?
Could be negative.
It'd be negative, it'd be pink, right? Because the safranin would bind just to the cell membrane and give that cell a red color. But if this were truly a gram-positive L form, then when it had its cell wall, it would look purple. So you could really drive yourself crazy if you had an L form, because sometimes they come up gram-positive, sometimes they come up gram-negative. I will guarantee none of you have L forms for your unknown. We would not do that to you. Another interesting wrinkle on cell wall structure is a genus of bacteria called Mycobacterium. Two of the more notorious, infamous species of Mycobacterium are Mycobacterium leprae and Mycobacterium tuberculosis. Obviously, causative agents of leprosy and TB. Mycobacterium has a cell wall that is filled with a waxy substance. That waxy substance is called mycolic acid. This waxy substance really helps to make these cells very resistant to a lot of things, including many chemical treatments. It tends to make that cell wall much less permeable to substances. That's one of the reasons why TB is so hard to treat. It's very hard to get the TB drugs into the cells. Another feature that makes TB hard to treat is it's got a very slow reproductive life cycle. If you have TB, you can treat it with antibiotics, but it sometimes takes a year or more of continued antibiotic treatment to effectively eradicate TB. That's a real problem. Well, there is a special kind of a stain which we will do called an acid-fast stain that really checks for the presence of this waxy cell wall filled structure associated with Mycobacterium. So these organisms tend to be very unique in being acid-fast positive. Most organisms that lack the mycolic acid in their cell wall are acid-fast negative. So this gives us a fairly rough and dirty test that's pretty easy to do to diagnose the presence of mycobacterial infections. When TB used to be a real problem in this country, it wasn't uncommon for someone who had signs of TB, you know, coughing, hacking up all kinds of stuff, to go into the doctor's office. Doctor takes a sputum sample, throws it on a slide, does an acid-fast stain, and if you get positive acid-fast results, that was pretty clearly Mycobacterium, and then more than likely, given the symptoms, probably the TB form.
What did the acid-fast check for?
It checks for the ability to retain this stain, and that is a function of whether you have this waxy substance in it. We'll talk more about the theory of this, John, when we actually do the acid-fast stain, but this is one we will do. And then finally, there are the weird and wonderful RK bacteria. Remember that, according to our currently accepted idea of classification, life is now grouped into 3 domains: eukarya, prokarya, and the archaea. The latter 2 are both prokaryotic. They were previously lumped together into the kingdom Monera in the 5-kingdom approach of classification. We really think that these were very early on a split off of the major evolutionary tree. And we'll spend some time talking about this group because they're a very interesting group in a number of ways, including their cell wall structure. Cell wall does not contain peptidoglycan. In some species of RK, the cell wall is made entirely of carbohydrate. In other cases, the cell wall is made entirely of proteins, no carbohydrate. So a tremendous amount of variation in terms of the cell wall structure, but it is not the peptidoglycan that we see associated with the prokarya domain. All the stuff we've been talking about at this point. So if you do a Gram stain on RK, what do they stain? They stain Gram negative. Okay? Because they lack the peptidoglycan layer, which is necessary to retain that crystal violet iodine complex. So all of these would be Gram negative. Okay. We will wrap up with cell structure on Monday. And then we will move into the dreaded section on taxonomy, which will be good because it will introduce you to Bergey's manual. And next week you're going to get your unknown, and eventually you'll have to use Bergey's manual. Okay? So, you ready to go for Dr. Morden tomorrow?
Yes.
That's fine, yeah, the lab will be open, yep. What do you need to do? Okay, the swabs are still out here, water's still out here. Yes, yeah, they're in there. You have your plates? There's extra plates in the bottom of the refrigerator if you ever need them. Okay. Yes, ma'am.
So, you know, those questions are end of month. Should we have it?
Well, definitely before the lab exam, but they're not collected and graded. No, that's up to you.
It's a study guide.
It's a study guide. Yep.
We didn't finish, and we can do that next week. We can finish that
Absolutely.
next week.
Absolutely. It's much better if you keep up with them,
Right.
but I do not collect them and grade them, and you don't have to do them, but you probably will suffer on the lab exam if you don't do them.
Yeah.
Thank you for your help and your emails. I got something yesterday. So
Great.
GMO emailed me. I was like, you knew me, so I was surprised, but thank you.
Yes, absolutely. Well, you've got lots to be proud of.
Yeah, I'm definitely pumped. I appreciate your help. Thank you.